So this is glucose. It's a 6-carbon carbohydrate that can either be in the linear form or it can convert to the cyclic form. And this is carbon 1, this is carbon 2, carbon 3, etc. And these aren't arbitrary numbers. This is officially carbon 1, officially carbon 2, etc. that you should just get familiar with. So when we have this linear form, how do we convert to the cyclic form? Well, to determine this, first remember this was carbon 5 with its hydroxyl, and remember this is carbon 1. Essentially, we can go through an intramolecular reaction where this oxygen with its lone pairs of electrons acts as a nucleophile, while this carbon acts as an electrophile. So we can go through an intramolecular reaction where this hydroxyl nucleophilically attacks this carbon number 1. And when it does that, it forms a bond, and when it forms that bond, it pushes these pi electrons up on this oxygen. And remember, we have free rotation around a lot of these bonds. So essentially what happens is this linear form will get in a very specific conformation. These bonds will bend in a very specific conformation. So now remember this hydroxyl for this carbon number 5, this hydroxyl, remember, nucleophilically attacks carbon 1. Remember we said this hydroxyl nucleophilically attacks carbon 1. So we see that here. It gets in this right particular conformation where we can go through the intramolecular nucleophilic reaction. So we nucleophilically attack, forming a bond, and when we form that bond, we push these pi electrons up on this oxygen. When we do that, we form this structure, where again, remember, we attacked forming a bond represented by this bond, and then when we did that, we push these pi electrons up on this oxygen, so then we would have that oxygen anion that would get protonated, forming this cyclic form. So now we form this cyclic form of glucose. So again, it's always on the carbon 5, that hydroxyl attacking, forming this cyclic form, and now we have the cyclic form. But this carbon 1, which again was originally the aldehyde, this is referred to as the anomeric carbon. And it's the carbon that was attacked by this oxygen, so now we have the cyclic form. But the point is, this carbon 1 is the anomeric carbon, and there are two types of anomers we can form. Because again, remember, this is an sp2 carbon. So because it's an sp2 carbon, it's planar. So therefore, because we have free rotation around this particular bond, this hydroxyl, this oxygen nucleophile can attack on this face, or it can attack on this face. So if we happen to have the specific rotation where we attack on this face, we form the beta anomer. However, if we attack on this face, we form the alpha anomer. So these are both glucose molecules. The only thing that differs is the stereochemistry of this carbon 1, this anomeric carbon. So again, this is the alpha anomer and this is the beta anomer. And the way I like to remember this is if the hydroxyl points away from this carbon 6, remember this is carbon 6, so if it points away, that's the alpha anomer, A for away. So if it points away from this carbon 6, that's the alpha anomer. However, if it points in the same direction as this carbon 6, remember this was carbon 6, so if it points in the same direction, it's the beta anomer. But the point is, depending on which side it attacks determines whether you form the beta anomer or the alpha anomer. But once we form these cyclic forms, we can go back to the linear form. And it's the same mechanism whether we're the beta anomer or the alpha anomer, so I'm just specifically going to focus on the alpha anomer. But we know once we form the cyclic form, we can go back to the linear form. So how do we do this? Well, the first step is we take this oxygen and we protonate it. So we simply protonate it. Now that we protonate it, we can go through a reaction where these lone pairs of electrons scooch down. When they fall down, they form a double bond. And when that double bond is formed, this bond breaks, and these electrons in this bond fall in the oxygen. And if we did that, we'd essentially form this structure. We'd form this structure, because again, remember, the electrons scooch down, forming a double bond, represented by this double bond, and then this bond breaks and these electrons fall in this oxygen, so we would get this guy. So now once we form this structure, we would essentially just simply deprotonate this oxygen. When we deprotonate this oxygen, now we have this aldehyde, which again, this is the linear form. It's in a very specific conformation here, but again, this essentially is the linear form. So that's the mechanism of how we go from the cyclic form to the linear form, and it would be the exact same mechanism whether it was the beta anomer or alpha anomer. But so we see these processes are reversible. We can have the linear form, then form maybe the alpha anomer, then go back to the linear form, then maybe the beta anomer, then back to the linear form, then maybe back to the beta anomer just by chance. But the point is, these processes are reversible. So this process of going from linear to cyclic to linear is referred to as motor rotation. But again, the point is, once we have the linear form, we can form the cyclic form, and there are two types. We can form the alpha anomer or the beta anomer.